So now we'll move on to part 2 of central nervous system development, which includes the higher centers of the brain. Now, what are the higher centers of the brain? Well, we have the cerebellum, which again we already talked about because it was derived from the rhombencephalon, but it really is one of the higher centers of the brain. And then the forebrain, or the former prosencephalon, that develops into the diencephalon and the telencephalon. Now, the diencephalon then goes on to form choroid plexus, as we talked about, forms within all the ventricles, what are called the circumventricular organs. They essentially secrete these sort of specialized chemicals and proteins into the cerebrospinal fluid. We won't go into them in detail, but that's their primary function. The diencephalon also goes on to form the pituitary gland, or at least part of it, the posterior lobe, and the optic vesicles. The telencephalon goes on to form the cerebral hemisphere, so those two large structures of the brain, and then the rhinencephalon, or nose brain, which are your olfactory bulbs and olfactory tracts. It also goes on to form the basal ganglia. So the diencephalon. What we have here are derivatives of the alar, or the dorsal plate. Again, sort of thinking similar to what we saw with the spinal cord development and then the brain stem. You don't actually get basal or ventral plates in the diencephalon, just the dorsal plates. But they go on to form the thalamus, the hypothalamus, and the epithalamus. Now, the thalamus and hypothalamus we talked about earlier in terms of what their functions are. The epithalamus then goes on to form the pineal gland, which it's not entirely well understood what it does, but it does have cells which produce melatonin, and specifically in response to low light levels. So melatonin, you probably heard, as a supplement you can take. Its primary function is to make you drowsy, and in the case of the pineal gland, it's secreted in response to low light levels. So it's probable that the pineal gland is responsible for making you sleepy when it gets dark. Now, the roof plate of the diencephalon will differentiate into the bilateral choroid plexus of the third ventricle. And again, the choroid plexus are going to make the cerebrospinal fluid. The pituitary gland. The pituitary gland is actually derived from two separate structures. The infundibulum, which is the floor of the third ventricle, and then the Rathke's pouch, which is actually an ectodermal derivative. It starts out sort of near the mouth and then migrates up towards the infundibulum, and then they merge together to form the pituitary gland. Now, interestingly, you can actually have remnants of the Rathke's pouch that persist. It's supposed to essentially sort of go away, the part that doesn't go on to form the pituitary, but it can persist. It can form a cyst, which is called a Rathke's cleft cyst. That actually doesn't always cause trouble, but it can get large, and if it does, it'll compress the neighboring structures, which in this case would be the optic nerve. So one of the primary symptoms of a Rathke's cleft cyst is vision loss. You can also get tumors that form from remnants of the Rathke's pouch that don't disappear as they should. And the tumor there is the craniopharyngioma. It is benign. It is not generally a cancerous tumor, but again, it can compress neighboring structures and cause trouble. Next up, the telencephalon. So with the telencephalon is what's going to go on to form the cerebral hemispheres. And so it starts by forming these two bulges. Now in this picture, you're sort of looking at the embryo from the side, so you're only seeing the one bulge. But there's one on either side, or there are supposed to be. I have some supplemental things for you to look at on the subject of something called holoprosencephaly, where you don't get proper division of the brain into two cerebral hemispheres. But in normal development, you get these two bulges that are going to become the cerebral hemispheres. They grow very quickly. The roofs of the cerebral hemispheres and the walls are going to become the cerebral cortex, that highly wrinkled exterior structure of the brain. The floors of the two cerebral hemispheres contain lots and lots of neurons, very densely packed neurons, and they're going to form the basal ganglia. Now, as the cerebral hemispheres are growing, they essentially fold over the diencephalon and then ultimately become continuous with it. So the meninges, the membranes that were separating those two structures, dissolve or dissipate, and the two structures are now essentially becoming one, or they become continuous with each other, which allows neural tissue from the thalamus to come into contact with the cerebral hemispheres and allows for that connection. The folding of the cerebral hemisphere starts at about embryonic month 4, when we get a cleft that forms in the wall, the lateral wall of each hemisphere. It's called the lateral cerebral fossa, if you're curious about what it's called. But essentially, it's going to define the temporal lobe of the brain, which is the first primary definition of the brain structure. Initially, before that happens, the fetal brain is smooth. And there's really only division between the two hemispheres. We don't have other divisions. But with the formation of the temporal lobe, we're starting to see basic brain structure setting up. And then by embryonic month 6, we actually have frontal, parietal, and occipital lobes in addition to the temporal lobe, all defined. And we're seeing folding on the surface of the cerebral cortex. Similar to what happens in the cerebellum, we get sort of lobules and then gyri that form, increasing the surface area dramatically, increasing the capability for neuron migration into that area dramatically. Now, on the inside of the brain, we should now have the 4 ventricles. So we talked about how they were derived from the interior of the neural tube and how they all contain choroid plexus. The final structure of the ventricles of the brain includes 2 lateral ventricles, one in each cerebral hemisphere, which are connected to the 3rd ventricle, which is within the diencephalon, which is then connected via the cerebral aqueduct to the 4th ventricle, which is in the rhombencephalon, which is then connected to the spinal cord, allowing cerebrospinal fluid to circulate throughout all 4 ventricles and then down into the spinal cord. Now, choroid plexus develops within each ventricle, and well, that's what's shown in the lower picture here. The choroid plexus is that sort of pink tree-looking structure that seems to be derived from the inside of the ventricles, and it's what's producing the cerebrospinal fluid. If you remember, back when we talked about trisomy 18, prenatally, sometimes you actually see cysts within the choroid plexus developing within the fetal brain. The choroid plexus is a structure that you don't necessarily see clearly, but you can see if there's a cyst there. That cyst would be cerebrospinal fluid that has not yet passed into the fetal system, likely because the fetal system is not quite ready for it, and then it sort of backs up into these cysts. It's associated with trisomy 18 because babies with that condition have it much more commonly than babies that don't have trisomy 18. But in most babies that have choroid plexus cysts, things are otherwise fine, and the cysts themselves do always drain. They don't damage other parts of the brain. And it's one of the reasons, it's sort of a nice thing to explain to families when you're talking about trisomy 18, because cyst on the brain sounds very, very bad. But when you understand what the choroid plexus actually is, and that it's really its only function is to produce this fluid, it all seems a little bit less scary. So the neuronal tissue of the cerebral hemispheres. First, let's define white matter and gray matter. White matter are the axons of your neuronal cells. Gray matter would be the cell bodies. And they just sort of, the names come from the color of the brain tissue. Now, the neurons of the brain originate from the ventricular layer, which would be the layer immediately adjacent to the ventricles, and then they migrate in waves towards the peripheral edge of the brain, or the superficial edge, so the exterior. The initial wave doesn't go very far, just sort of goes out just a little bit, and then each successive wave will migrate a little bit further, so that you're building the neurons from the deeper tissue layers, the interior of the brain, outward to the periphery. Once all of those waves of neuronal migration are complete, then you see support cells forming in those areas and sort of migrating outwards as the neurons did. Now, who is this man and why is he on here? I'm really hoping that some of you know who this is, but just in case you don't, this is David Suchet playing Hercule Poirot from Agatha Christie's famous novels. Just fantastic videos produced of these novels. Clearly we've wandered off topic, but Poirot was very fond of talking about his little gray cells and how that helps him to solve all of his murder mysteries. And if you haven't watched it, I mean, I really can't require you to watch it, but I think you would enjoy it because they're all fantastic.
And at any rate, we will move along here. Okay, so cerebral hemispheres. We talked about how the cerebral hemispheres divide. Now we need to talk about how they come together, how they talk. There are actually several commissures that allow the cerebral hemispheres to communicate with one another. We're really just going to focus on one, which is the corpus callosum. So it actually runs the entire length of the cerebral hemispheres, allowing connection across the entire length. Nerves run across the corpus callosum to allow for that to happen. And there can actually be absent, either partially or entirely, with extremely variable effects. To the degree where there are going to be some people who are missing the corpus callosum who function very normally, others that have intellectual disability, some that have seizures, some that are very impacted with very restricted function. What would that look like? So these are MRI scans. Here's the caveat, I don't read MRI scans, but I think this part is at least fairly obvious. So in the first picture, in the blue circle, that sort of C-shaped structure there is the corpus callosum on an MRI. And then here is where it should be on this MRI scan and does not exist. And that second image came from a child who had intellectual disability, who had seizures, and was receiving an MRI as a workup for those things. And certainly the absence of the corpus callosum would explain that. So back to our summary table. So from the prosencephalon, we get diencephalon, which then remains diencephalon. It's actually the only part of the brain that retains its embryonic name. And then also the third ventricle. And then also from the prosencephalon, we get the telencephalon, which forms the cerebrum and the lateral